This presentation is about the receptors of the body. First, a sensation is the information carried by a sensory pathway. A perception is when that sensation reaches the sensory cortex of the brain and you now have a conscious awareness of that sensation. Many people use these terms interchangeably, but they usually are referring to perceptions. The senses can be divided into general senses and special senses. The general senses include temperature, pain, touch, pressure, vibration, and proprioception. What makes them a general sense is that the receptors are located throughout the body. The special senses are sight, smell, taste, hearing, and equilibrium or balance. What makes them different from a general sense is that there are special receptors in only one special organ for each sense. For example, you only see with the photoreceptors in the eye. You only hear with receptors in the inner ear. This presentation deals with the general senses only. This image is showing what is called the receptive field, or the area monitored by a single receptor cell. If the receptive field is large, then the sensation is very general. If the receptive field is small, the sensation is more precise. Let's look at this example. Here we have two small receptive fields, and these pins that I have right here are pointing in two different receptive fields, and so the individual would feel two separate pinpricks. And the skin that has this small receptive field, for example, would be the skin of the fingertips. Same section of skin, the pins are the same distance apart, but because the receptive field is large, both of the pins are in the same receptive field. That means that the person will actually perceive only one pinprick. And this would be in locations where sensation is not really important or as important. So let's say the skin of the trunk, the back, the legs, for example. The nervous system transmits information through the neurons by electrical signals. So how does a sensation such as touch or taste turn into an electrical signal? This process of converting a stimulus into a change in membrane potential is called transduction. This process changes a little bit depending on the type of receptors. And then the signal travels through what is called a labeled line, or a group of axons that carry only one type of stimulus. So the sense of touch only travels along axons for touch, and sight only travels over axons for sight. Sorry. The receptors in the body are classified according to their function, or the type of sensation that is carried. There are nociceptors, thermoreceptors, chemoreceptors, and mechanoreceptors. Nociceptors are receptors that only respond to pain. There are two types of pain and pain fibers. Type A fibers carry fast pain signals, and type C fibers carry slow pain signals. When a person describes their pain, they usually use terms such as sharp and shooting or dull and achy. The sharp shooting pain is the fast pain carried over type A fibers, and the dull achy pain is slow pain carried over those type C fibers. Another type of receptor is a thermoreceptor. Like a thermometer measures temperature, a thermoreceptor detects changes in temperature. Chemoreceptors respond to chemicals in the body fluids. These chemicals could be ions, proteins, or even hormones. Mechanoreceptors respond to changes that distort the plasma membrane. There are several types of mechanoreceptors: proprioceptors, baroreceptors, and tactile receptors. Proprioceptors monitor the position of joints and muscles. Baroreceptors respond to changes in pressure, usually blood pressure, and tactile receptors respond to touch. Another way to classify receptors is to consider when they are active. There are tonic and phasic receptors. Tonic receptors monitor the presence and intensity of a stimulus. They are always active, even in the absence of a stimulus, and increase the number of action potentials they generate when a stimulus is applied. And they will continue to send action potentials as long as the stimulus is present. Some examples of these tonic receptors are photoreceptors and mechanoreceptors. And as we see in this image here, there is no stimulus, but we still have action potentials. Now here we have a stimulus, and as long as the stimulus is present, we have an increase in the number of action potentials. And when the stimulus goes away, then we have a lower number of action potentials, but there are still action potentials when there is no stimulus. That is different from a phasic receptor. Phasic receptors also respond to changes in stimuli, but they are normally inactive. When a stimulus is first applied, they begin to generate action potentials. But the longer the stimulus continues, the receptor will stop generating action potentials. This process is called adaptation. Some examples of this include touch and pressure. And so in our picture here, we have normal or no stimulus, and then we have an increased stimulus. A stimulus, and then no stimulus here. And so we notice that in the time when there is no stimulus, there are no action potentials on either end. And then during the time that we have a stimulus, at the beginning, there are action potentials, but as it continues to be stimulated, there are no more action potentials here. And then as it drops, then there are action potentials telling the body that there is a drop in stimulus again. So I mentioned adaptation. Adaptation is the reduction in sensitivity in the presence of a constant stimulus. This can be either peripheral adaptation, when the receptor stops responding to the stimulus, as we mentioned in the slide about phasic receptors, or it can be central adaptation, meaning that the receptors are still generating action potentials and sending information to the central nervous system, but the central nervous system stops responding to that stimulus. An example of peripheral adaptation and the phasic receptors is the sensation of touch. Think about the fact that your clothes are touching your skin all day, but do you constantly feel them? Or is it only when they move against your skin? Same thing with your hair. Unless your hair is moved by something, you probably don't notice it against your skin. An example of central adaptation is the sense of smell. Have you ever walked into a room and thought that it smelled bad, but after some time you don't notice the smell anymore? Or if you wear perfume or cologne, you don't smell your own perfume after a while. That is because the central nervous system is not responding to the olfactory signals that are being sent to the brain. Here we begin discussing several of the specific types of general sensory receptors. We will discuss free nerve endings, the root hair plexus, tactile discs and corpuscles, lamellated or Pacinian corpuscles, and Ruffini corpuscles. Free nerve endings are the most general of all receptors. They extend up into the epidermis and respond to tactile, pain, and temperature stimuli. The root hair plexus monitors distortion of the body surface, specifically movement of each hair follicle. This is why you can tell if even one hair has been moved. Tactile discs and Merkel cells are always associated with each other. They are also located in the epidermis. The Merkel cells are large epithelial cells and are attached to the basement membrane of each cell is the tactile disc. They have very small receptive fields and respond to fine touch and pressure. The tactile corpuscle is in the upper portion of the dermis. Notice that there is a connective tissue capsule surrounding the dendrites of this sensory neuron. These respond to fine touch and pressure stimuli, as well as some vibration. The lamellated corpuscle, or Pacinian corpuscle, is located deep within the dermis, almost to the hypodermis. So they respond to deep pressure. They have layers of connective tissue around the dendrite, giving them the name lamellated. And the last type of general sensory receptor is the Ruffini corpuscle. Again, they are located deep in the dermis and are sensitive to deep pressure.